So nucleic acid, there is two different types. So there is ribonucleic acid. Since this is in English, so that's quite easy to understand. This is R, this is N, this is A. So that's RNA. The other one is supposed to be deoxy. The more important one is deoxyribonucleic acid. I'm sure you've heard the name RNA, DNA before, but okay, I'm sure you've heard the name RNA and DNA before. Just that you may have not seen the full name of it, or maybe already learned in PDT. I'm not sure. But this is what you need to know. For Form 5, you need to know the full name. We have RNA, and we also have one more by the name of DNA. That's quite obvious. DNA. So that's the full name. Both RNA and DNA, are supposed to be polynucleotide. Nucleic acid, RNA, DNA, I already explained this in the last class. Both of them are polynucleotide. The word poly means many. So there is many nucleotide, and because there's many nucleotide linked to one another, they form a chain, satu rantai. So because it has many nucleotide, the basic unit bahan asas, the basic unit monomer for nucleic acid is known as nucleotide. So the basic unit is nucleotide. So when we start this nucleic acid explanation, the first thing we have to do is we have to describe what is the monomer, what is the structure of the monomer. The monomer is nucleotide. How does it look like? Because you also need to know how to draw a nucleotide. So let's see how a nucleotide looks like. So before that, just to re-explain what we mean by polynucleotide. This one here, the circle, the red circle represents a certain amino acid. And this square here, the blue square, represents another different amino acid. If you remember what you have already learned in class, there is 20 different types of amino acid, diplogenous amino acid. Maybe if the Kepung student haven't learned this, there is 20 different types of amino acid, diplogenous. And how do we show that there is 20 different types of amino? How do we show it's different amino acid? That is why we are using different shape. By using bentuk, shape, different shape, we are trying to show you is different amino acid. So if you have two amino acids side by side, an enzyme can catalyze the reaction. They can form a bond. And the name of the bond is peptide bond. In our chapter biochemical, this chapter 4, every one of them, carbohydrate, lipid, protein, nucleic acid, all of them have bond. But syllabus wise, the bond in the protein, you must know the name, which is peptide bond. By right, carbohydrate also have bond. They have a name, but no need to know in our syllabus. Same goes with lipid. Lipid between glycerol, fatty acid, also there is a bond. Even though I'm not giving the name of the bond in bio explanation, later you'll be learning the same thing in chemistry. In chemistry, they'll give you the name. But for bio, if you don't know the name of the bond, never mind. But for protein, you must know the name of the bond, which is peptide bond. So dual amino acid, two amino acid linked to one another, they form peptide bond. What you get is now known as a dipeptide. If I have more amino acids, and I'm using all of these different shapes to show you it's different amino acid. When I have many amino acid, we get a polypeptide. So this is a polypeptide. Many amino acid. Many amino acids linked to one another through peptide bond. That's a polypeptide. Many amino acids linked by peptide bonds to form a polypeptide. So that's protein. Now we're learning RNA, DNA, nucleic acid. So nucleic acid, if you want to make polypeptide, you need amino acid. That's the monomer. If I want to make DNA or RNA, I want to make nucleic acid, I need nucleotide, which is the monomer. So I'm just using this shape to represent a certain nucleotide. If I have another nucleotide, they can also form a link. They can also form a link. There'll be some reactions, condensation reaction, and these two are already linked. If I have more nucleotide, I can link them together. Now I have many nucleotide linked to one another. We call this polynucleotide. Banyak nucleotide berhubung antara satu sama lain. Many nucleotide linked to one another, but in here you don't have to know the name of the bond. Polypeptide, you need the bond. In here, yes, they have a bond, the name not needed. So we have many nucleotides linked to one another through bonds. You get a chain. You dapat satu rantai. It's a chain, and that chain has many nucleotides linked to one another. So this is a polynucleotide. RNA and DNA are both polynucleotide, meaning both RNA and DNA are chains of nucleotide. So just to explain to you the meaning of polynucleotide, nucleotide linked to one another through certain type of bond. Okay, fine. This is how they look like, but now we go a bit more specific. Before I can make the polynucleotide, I need to describe to you how does each nucleotide looks like. So that will be the first question we can ask you. Nucleotide. Every nucleotide consists of a sugar known as pentose sugar. We call it pentose sugar not because they have pentagon shape. It is known as pentose sugar because it is a 5-carbon atom sugar. Inside this sugar, there is 5 carbon atom. 5 is penta. So that's why we have pentose sugar. So every nucleotide has a pentose sugar. It's known as pentose because they have 5 carbon. If you remember glucose has 6 carbon. This one has 5 carbon. We call it pentose sugar. This pentose sugar will be linked to what we call as a phosphate group. This is nothing new. Already know ATP, energy molecule, it has 3 phosphate group. When we explain about active transport, one of the phosphate group will attach to the carrier protein. If you remember, the carrier protein shape changes after the phosphate group attached to the carrier protein. So this is not new. And to show its phosphate group, inside the circle, we'll add an alphabet P, capital P, to represent as a phosphate group. The phosphate group is linked to the pentose sugar because I'm using a straight line here, meaning this is a covalent bond. So this bond is every time you see a straight line, it's a covalent bond, which you'll be learning in your chemistry also. So it's a covalent bond. It's not new because under lipid, I already show you like carbon needs to have 4 covalent bond, oxygen needs 2 covalent bond, hydrogen needs 1 covalent bond. To represent covalent bond, we always use a straight line. So the phosphate group is attached covalently with a pentose sugar. The same pentose sugar is also attached to what we call as a nitrogenous base. So nitrogenous base actually got 5 different types. Tadi when I start this nucleic acid, I told you nucleic acid is nucleic acid is one step higher than the other chemicals that you're learning in this chapter, mainly because of this nitrogenous base. Nitrogenous base, there is 5 different types, and the 5 different types have weird names or unique names. That you might find a bit hard to remember the names of the nitrogenous base. I'll show you the name later. For the moment, this is one nucleotide. You must be able to draw a nucleotide. There are some partial questions that we ask you to draw, and we don't ask you to draw in a essay question. We ask you to draw in structure question, compulsory question, maybe 2 marks or maybe 3 marks. So you are supposed to know how to draw a nucleotide. We'll draw that later in the notes. This is one nucleotide, basic unit of nucleic acid. If you are describing a nucleotide, some of you may mention nucleotide has a phosphate group. That phosphate group is linked to pentose sugar. The pentose sugar is linked to nitrogenous base. Maybe your explanation is in this sequence. From here, you explain here. Preferably for when you describe them, if you need to describe nucleotide, it will be better if you start from the pentose sugar. You mentioned it has a pentose sugar. That pentose sugar is linked to phosphate group. That same pentose sugar is also linked to nitrogenous base. Better for you to explain from the pentose sugar, like what I did also. When I explain about, when I start to describe this, I started first with pentose sugar. I introduce you pentose sugar. Then I mentioned there is phosphate group. And that phosphate group is linked to pentose sugar. Then I introduce to you nitrogenous base. Then I mention it is attached to pentose sugar. So that's how that is a proper way of describing nucleotide. You start with pentose sugar and then you describe the two other things that is attached to the pentose sugar. That will be a better organized answer. So in case if they ask you to describe nucleotide, start with this, explain this, then explain this. But more important for your exam is you must be able to draw this. So if you check your notes, now I'm going to your notes. So in here, the simplified looks like this. What I have in the notes is a bit more detail. So if I go here, this is the nucleotide that you have in your notes. Okay, one of the students asked me, what is the A? I haven't come to that. I told you that nitrogenous base got 5 different types. The A is one of the type, which, like what Mian said, is adenine. I'll come to that. I'll explain what is A and onward. There's 5 different symbols. Okay, so here, a bit more detailed diagram. You should be able to identify phosphate group. Why is this a phosphate group? Because it has a phosphate atom. It is linked to this, which is the pentose sugar. Pentose sugar. This pentose sugar has 1, 2, 3, 4, 5. It has 5 carbon. That is why we call pentose sugar. This is a nitrogenous base. Like what your friend has said earlier, this is adenine. The symbol for adenine is A. That is what you saw earlier. You can clearly see in here why we call it nitrogenous base. 1, 2, 3, 4, 5. The nitrogenous bases will have a lot of nitrogen. That is why they have a name nitrogenous base, just to let you know. But this diagram here, this diagram here is too detailed for your level. I'm just showing you how they look like. You need to know the shape that looks like this. As long as you know this is phosphate group, this is pentose sugar, this is nitrogenous base, enough. We need to draw that in our notes. So we are going to draw the nucleotide in this empty space here. So how do you draw a nucleotide? You start with the phosphate, the complete circle. Inside you label, you can copy as I'm drawing, you can also do this. So we start with a circle, which is this circle, phosphate group. Inside you add in a B. So if I were to add in my, okay, it should look something like this. Okay, phosphate group. The trick is, after you redraw the from here, I'm going to draw a line slightly slanted like this. Okay, so earlier there was problem with internet connection, so some of you maybe my voice was muted. I repeat. So this is the phosphate group. You draw a circle, extend a line downward, slanted, not a straight. It's a straight line, but slanted straight line, similar to what we see here. This one. After you already draw this straight line, use this straight line to draw the pentose sugar. So somewhere from here, make it into a pentose sugar. So you draw a pentagon shape like this. So the best way to do it is use this line to become a pentose sugar. And from here, you extend one line, and at the ending of this line, you need to draw a nitrogenous base. And the nitrogenous base that we are drawing is long and the ending is pointed. Inside here, you're supposed to write alphabet A, like what your friend asked here. Why you have an A inside here? So, okay. So this is how a nucleotide should look like. you should be able to label this diagram. Okay, you should be able to label this as phosphate group. Okay, so you can whatever that I'm adding here, you can also add into your notes. So I maybe make it a blue color, for example. Okay, maybe a bit smaller also. Okay, too small. So this is the phosphate group. Also label this one as, and also label the last one here as the pentose sugar. So label this as the pentose sugar. Okay, so hopefully you already draw the diagram. So take note, the diagram is 1 mark, the label is 1 mark. The diagram is 1 mark, the label is 1 mark. Whenever we ask you, in my normal classes also that we have in the center, I always insist on drawing certain diagrams because we ask that in exam. Whatever that you're drawing in class, in my class, can come out in the exam. So like if you see, this is the diagram you have done. Earlier in the WhatsApp group, I've sent a message. I also asked you to download one of the past year question, the recent question year 2018. So if you look at that question, year 2018. So this is the question that I asked you to download. Hopefully you already download. So this is 2018 question. It's actually about organelle, but part of it was the DNA question. Question A and question B is something to do with organelles. By now you have done your revision. All of this is easy. You can easily identify P is the cell wall, Q is the vacuole, R. Okay, if you look at the S is pointing to, I think, RER, rough endoplasmic reticulum. This R here is pointing to the nucleus, but inside the nucleus there is a lot of black, some random lines. That is supposed to be the chromosome. So in this question here, they ask you about, if you look at this question, they ask you what is R. So R is the chromosome, S is the rough okay, never mind. You can write R as nucleus and S is the rough endoplasmic reticulum. What is the main component which forms the R? So what is the main component inside the nucleus? Inside the nucleus we have chromosome. The next page question is, take note, inside the chromosome we have DNA. But you see the next page question, draw and label the basic unit structure of the main component you name in 1B1. So in 1B1 you may have labeled chromosome, correct? If you label DNA also correct. So in case you label DNA, what is the basic unit? The basic unit is this. So this is what you need to draw. This is what you need to label. The drawing is 1 mark. The labels are worth another 1 mark. That is the 2 marks for this question. So it's a compulsory question in exam. Actual SPM question. You need to draw in here phosphate group. You need to draw a pentose sugar like this, and you need to draw one of the nitrogenous bases. My preference, I always like to draw the one that is pointed like this. You should have a P inside here, and you should have a A inside here, or there is another few choices. Later I'll explain. This is 1 mark. The label, phosphate group, pentose sugar, nitrogenous base is worth another 1 mark. So kadang-kadang kita suruh you lukis. This is a question that you need to draw the nucleotide. Okay, coming back to this. So this is the question. I'll come back to the question later. I'm going back to your notes. So we are now somewhere here. I think no. Okay, so this is what we were doing earlier. Okay, so far I have given you, one of the students asked, doesn't the DNA is double helix? Yes, DNA is double helix. I haven't come to that yet. I am still introducing what is a nucleotide. We haven't even gone into RNA and DNA. We are just introducing to you the basic unit. After you know what's the basic unit, only then we can go to the complex one. So I'm still at the beginning of it. the basic information. I haven't make it complex yet. For the moment, bahan asas. We'll go one by one into the double helix. Okay. So far, I have, so if you look at your notes here, you should be able to understand some of the points here, which is nucleic acid monomer is nucleotide. Now you know what is it. Each nucleotide has a pentose sugar. Now you know. It has a phosphate group attached to pentose sugar. You know. It has a nitrogenous base attached to the pentose sugar. Also you know. This sentence ribose, deoxyribose, saya belum explain. Buat masa ini, you tahu nucleotide ada pentose sugar, phosphate group, nitrogenous base, and how do you draw them? This is how you draw them. Any question? If you have a question, you can ask me in the chat group. Don't know question, just press the no button so I can move to the next explanation. There's some other questions here, which is someone asked me earlier. Okay, Surya Kumar, you have asked about ribonucleotide, deoxyribonucleotide. I repeat, I haven't come to that. I'm still at the beginning. What is nucleotide? I'll come to ribonucleotide, deoxyribonucleotide after this. I've only explained what I have underlined here on the screen. I haven't come to that. Deoxy and ribonucleotide, I haven't come to that. And another student asked me a question. What happens to the pentose sugar when it's not attached to the nitrogenous base? It could be used to produce energy because sugar is a sugar. It can be used to produce energy, but our body's choice is always glucose to produce energy. If they are not used, most probably they can be converted into, it's interconvertible, meaning we can take carbohydrate, we can change it into a lipid. We can take a lipid, we can change it into protein. Your liver can do all of that. So if there is a situation where there is too much of pentose sugar, you can change it into something else that can be used by the cell. The cell or the liver can do that process. But normally, what you mentioned, what happens if pentose sugar is not attached, generally doesn't happen. If it happens also, our body can convert it into something else that can be used by the cell. Nothing will go to waste. They can convert it into something else, they'll use it. Answer for the I think Raj Wilson's question. Okay, I'll come back to this. If you don't have any question, I'll come back here. So you know what is nucleotide. Now let's make it a bit more detailed. So we go back to this picture here. So if you are drawing a nucleotide, start with use a different color. Start with a circle to represent phosphate group, extend one line down, use that line to form a pentose sugar, use the line to form a pentose sugar, and from here form a nitrogenous base. Someone asked me earlier what is A, I'll explain later. Okay, so that is how nucleotide looks like. What I didn't mention earlier is when you draw this, you cannot draw this line from here. The line must come from the corner because this is where the carbon is located. It must come from the corner. In case you drew them from here or even here, that's wrong, not accepted. Dia mesti daripada corner. Same thing with this line here. You cannot draw from here or from here. It must come from the corner. In chemistry, the corner is where the carbon atom is located. So the link must be to the carbon atom. Make sure when you draw, the line is from the corner. The line is from the corner. That is something I forgot to mention earlier. So check back your diagram that you already draw in your note. Take note of this. The line must be from the corner. Straight line, but it's coming from the corner. In Malay, we say bucu. The pointed part, that's where the line will come from. Okay, we are done with nucleotide. So nucleotide is the monomer for nucleic acid. Now we are going to make it a bit more detailed. So pentose sugar, there is two types. One is known as ribose. Another one is known as deoxyribose. So pentose sugar ada dua jenis. Ribose dan deoxyribose. Just to show you is different, I'm using different color. Ribose, deoxyribose, there is two types of pentose sugar. Based on that, This is a nucleotide. If you are explaining about nucleic acid, then this sugar is pentose sugar. Kalau nucleic acid, if you're explaining nucleic acid, this is a pentose sugar, you give a general name. If I tell you that this is ribose, kalau ini ribose, this whole thing becomes ribonucleotide. You maintain the name nucleotide, but you make it specific by adding the name of the sugar at the front, it becomes ribonucleotide. We use ribonucleotide to make RNA, meaning ribonucleotide is the monomer for RNA. Untuk buat RNA, kita perlukan ribonucleotide. Ribonucleotide looks like this, but just that the sugar is specific, it's ribose sugar. If you want to make nucleic acid, you use pentose sugar, general name. If you want to make RNA, specific name, we need specific nucleotide by the name of ribonucleotide. If you want to make DNA, okay, if this is deoxyribose sugar, then what should be the name I write here? You answer to me privately, what's the name I should write here? Okay, good. All of you are giving me the answer for this. So what you have here is deoxyribonucleotide. If you have deoxyribonucleotide, you can use this to make monomer, to make DNA. Monomer for DNA, deoxyribonucleotide. Monomer for RNA is ribonucleotide. What's the difference between deoxyribonucleotide and ribonucleotide? It's just the sugar. This one has ribose, this one has deoxyribose. This one and this one, both of them are nucleotide, general name. Specific name, ribonucleotide, because they have ribose sugar. Deoxyribonucleotide, because they have deoxyribose sugar. Both of these pentose sugar. Both of these are pentose. I think Leandra's question was that. When does it become ribose? When it becomes deoxyribose? Both of these pentose sugar. It all depends on what the cell wants to make. If they want to make a RNA, they'll use a ribose sugar to make this. If they use a ribose sugar, it becomes ribonucleotide, who can use it to make RNA. If the cell wants to make DNA, they don't use ribose sugar, they use deoxyribose sugar to form this. So you can use this to make DNA. To make nucleic acid, you need, if you want to make nucleic acid, the monomer is nucleotide. In the nucleotide, what you have a pentose sugar. So you nak buat nucleic acid, you perlu nucleotide. Dalam nucleotide ada pentose sugar. I think it's obvious now. But if I want to make RNA, I need to have a specific one by the name of ribonucleotide, which has a ribose sugar here. If I want to make DNA, I need deoxyribonucleotide that has a deoxyribose sugar here. And another student asked me, how do we differentiate whether it's ribose or deoxyribose? Is it based on the color? No. Your exam question will be in black and white. You won't have a color there. I'm using the yellow color and green color to show you is different. Just to make it easy for you to understand, it's something different. If you see the phosphate, I use same color. This one I use the same color, but only these two I change the color to show you is different. How you're going to know is different in the question is based on what they're asking in the question. Whether they're asking you about RNA or whether you're asking you about DNA. That's based on the question. Whether they want about RNA or DNA, you can decide whether you need to use this or this. But I'm making this statement, listen carefully. Untuk exam, SPM will ask you to draw nucleotide. Will ask you to draw nucleotide. Why I'm giving you this is sometimes in the exam question, they can ask you, what is the difference between DNA and RNA? Apa beza DNA RNA? If you want to answer that question, what's the difference between DNA and RNA? The main difference is monomer for RNA is ribonucleotide. Monomer for DNA is deoxyribonucleotide. So I'm explaining this because sometimes the question is, what's the difference DNA and RNA? One of the main difference is the basic monomer. Cine-ribonucleotide, cine-deoxyribonucleotide. So the student that asked me the question, Prashina, I think. You know the difference based on the question, based on what they want. Is it DNA or RNA? There is another way to know the difference. You'll understand that when I start to explain about nitrogenous base. Buat masa ini, I haven't come to this yet. Once I come to this, there's another way to differentiate. Later you'll see. So I need all of you to do something now. Go back to your notes here and I'll clear all the. Okay. So in here, right at the bottom of this page, page 5, you see these two names, deoxyribonucleic acid and also ribonucleic acid. So if possible, add the name that you saw earlier. So not enough space, maybe you can write on this side. So deoxyribonucleotide. Deoxyribonucleotide dash monomer. For some reason I like red color, so I'll use red color. Okay, so I need you to add this. Maybe you just put an arrow here. Deoxyribonucleic acid. The monomer is deoxyribonucleotide. Deoxyribonucleic acid, DNA. The monomer is deoxyribonucleotide. The monomer for ribonucleotide, ribonucleic acid RNA is much easier. I just have to remove the word deoxy from the answer that I have on top. So ribonucleotide is the monomer for ribonucleic acid. Maybe you can just add somewhere in that page, preferably somewhere. If the question is, what's the difference DNA and RNA? One of the answers that you must give is the monomer. And the monomer for DNA is deoxyribonucleotide. The monomer for RNA is ribonucleotide. The monomer for nucleic acid is nucleotide. No need ribo, no need deoxy. Nucleotide is for nucleic acid. General answer. DNA, deoxyribonucleotide. RNA, ribonucleotide. Any question? Okay, so from nucleotide, we went into ribonucleotide, deoxyribonucleotide. Now is the time for us to explain about the nitrogenous base. So that will be the next explanation. And that is the most complex explanation, I think, for this. So I hope you don't have any question on the diagram that you can see now on the screen. So now we are going to this. Someone already asked me earlier, so what is this A? Okay, so now nitrogenous base, there is 5 different types. Nitrogenous base got 5 different types. The problem is the name of that 5. That makes this thing a bit complex. The first one is adenine. You need to know the name. Adenine. The second one is guanine. Third one, thymine. Fourth one, cytosine. And the last one is uracil. Sorry, you need to know all the names. And not only you need to know the name, you need to know the shape also. You can see the color is not important, the shape is important. I'm just using color to show you it's different and just to make the presentation attractive. Adenine, guanine are longer, these 3 is shorter. Both of these that is longer, the length is same. Sama panjang, but the ending here is pointed for adenine. For guanine is rounded. Thymine, cytosine, uracil are all 3 shorter, but these 3 same size, but they are shorter compared to these 2. And thymine pointed inwards, cytosine rounded inwards. Uracil has the same shape with thymine. But what happens is thymine is only found in DNA. You can only find thymine in DNA. Uracil you can only find in RNA. Adenine, guanine, cytosine you can find in DNA and RNA. Only thymine. That's why thymine, uracil same shape, because they are not found together. DNA has thymine, RNA has uracil. The reason we use shapes like this is to show, if I asked you, this adenine can pair with another one, you think it can pair with what? That means I can take another one of this and I can attach it here. Which one you think can pair with adenine? You can give me the answer in the chat group. Good. So I don't have to explain, you automatically, so all of you are giving me the right answer. You automatically can see the importance of this pointed and rounded. Good, all of you are giving me the answer. We use pointed and pointed inwards to show adenine and thymine. If not, adenine and uracil is a pair. You can attach them together because they have complementary shape, pointed and pointed inwards. Same thing, that means adenine is a pair with thymine, or it could be a pair with uracil. How are we showing you they are a pair? By using this specific shape. Guanine is a pair with cytosine. Meaning I take this cytosine which has a rounded shape here and I bring it here. They can't attach because this is pointed, this is rounded. But I bring this here, they can attach complementarily. In Malay, sepadan, because bentuk dia sepadan. In English, the word is complementarily. I'm very sorry, you need to know the 5 names in the syllabus and you also need to know the shape. So if you refer to your notes, the names are already there. The names are already there. You can see the names here. You can see the word adenine, guanine. I'll make it into this. Okay, you can see adenine, you can see guanine, cytosine, thymine, uracil. All the 5 names are here, but the shape is not given. So I need you to draw all the 5 shapes, maybe somewhere at the side here, or maybe somewhere here, or any other space, maybe at the top here also. Somewhere, find some space, or even in the cover, preferably in this page, I think on this side. You are supposed to know the shape. The shape is universal, meaning anywhere in the world you go, if you're explaining about adenine, this is the shape. This is like the biology scientific rule. When you draw adenine, this is how you must draw adenine. Guanine is this. These are the 3. Please draw, and once you're done, just put a tick yes in the participant list, so you can. So I know that you are done. I can move on to the next explanation. In between, if you have a question, you can ask. I'm happy that most of you have answered my question earlier, which is thymine and uracil. If you have done copying, you don't have to do any shading. You want to do any coloring, you do that later. For the moment, what you need to do is just draw the basic shape, meaning just draw like this. Should be fine. Like just the basic shape enough. You can use a ruler for the straight lines. Like that enough. For cytosine is like this. With the ruler, it becomes the main thing we want to see is the ending. And I repeat, adenine, thymine, longer, both same length. Thymine, uracil, cytosine, same length. They are shorter compared to the other 2. There is a reason why they're longer and shorter, but we are not giving it in Form 5. No need to be artistic. I repeat, the shape, the ending, and the beginning. The ending is important. Whether it's pointed or rounded, or pointed inwards or rounded inwards. I'll take on average, so around 60, more than 60 of you already put a note. So, okay, so I'll consider that is done. Okay, so if you have questions, please ask in the chat group. We'll move to the next. So now you know the 5 types of nitrogenous bases, but thymine only in DNA, uracil only in RNA. Fine. So I have completed nucleotide. So now we can go to RNA and DNA. So we are now explaining how does the RNA looks like. This is one nucleotide. But because I want to make a RNA, This nucleotide, if I want to be specific, because nucleotide is to make nucleic acid. Kalau saya nak buat RNA, saya perlukan wait, there should be a name here. Okay, this is nucleotide. If I want to make a RNA, I need this is a ribonucleotide. Nucleotide is for nucleic acid. We are making RNA, we need ribonucleotide. So this is one ribonucleotide. What I'm doing now is I'm adding in one more ribonucleotide. They form a link, covalent bond. I add another ribonucleotide. So now, and the covalent bond already formed. I now have a chain, satu rantai, a chain of ribonucleotide or a chain of nucleotide. This chain of nucleotide, satu rantai, single chain, is RNA. RNA is single strand. ST, the word we use is strand. STRAND. Strand means rantai. So RNA is single strand. I am asking you a question. How am I showing the examiner that this is RNA, not DNA? And give me the answer privately. How am I showing the examiner ini bukan DNA, ini RNA? The answer single strand, no, because DNA is double strand, yes. But when the double strand separate, they can also become single strand. My question is, in this diagram, how am I showing the examiner that this is RNA? It cannot be a DNA based on my diagram here. Okay, very happy to see all of you giving me the answer, which is, it has Kalpana, this is okay, some of you answering the word it's a single strand. Yes, this is a single strand, but DNA is double strand. Yes, the double strand is made actually of two single strand, meaning DNA pun ada single strand, tapi dia ada dua single strand yang bergabung, they bind. So my question again, specifically in this diagram, how am I showing it's a RNA? By using this. You can't find this in DNA. So I'm happy that almost all of you have given me the answer, uracil. Very good. You deserve a clap. And my next question, which you need to answer, which is, can I draw this RNA by having uracil here and having uracil here? All the three places I use uracil. Instead of having, like in here, you have adenine, guanine, uracil. Can I make all three uracil? Can I make all the three uracil? Many students write yes. Many students write no. And there are some students very smart. Yes, question mark. That means they are not answering the question, like they're asking me back. No question mark. So that are the smart students who don't want to be trapped with the wrong answer. And there's one student who just blindly say, I don't know, sir. Okay, fine. Okay, if you say don't know, good, because I haven't explained the answer. And I'm not surprised that you don't know. Okay, what happens here is a normal RNA will have thousands of nucleotides. Thousands. I draw only three because of limited space, and it's enough for you to understand. But a real RNA will have thousands of nucleotides. Thousands. And what nitrogenous base they have is random. You can have all the three uracil. You can even have hundreds of uracil in a row. Can, because it's supposed to be random. You draw all three uracil, correct. You draw all three guanine, correct. You draw all three cytosine or adenine, correct. The rule is, when you draw RNA, you cannot have thymine. That's the rule. You cannot have thymine. The moment you have thymine, that's already wrong, because thymine is for DNA. So that's the rule. Tak boleh ada thymine. So the others you can have. You want to make all the three adenine, also can. No problem. But a general rule is, we are drawing this to explain to someone else what is a RNA. So it will be better, instead of writing all the three U's, we also add a adenine, we also add a guanine to inform the other person who is looking at this diagram. Oh, adenine also can. guanine also can, uracil also can. We make it a point to show all the possibilities. But from all the possibilities, what you must have is this one. Uracil, one of it must be uracil. That's the rule. You cannot have thymine, you must have uracil. The others, we make this diagram better by informing the examiner, look, I put a adenine, meaning RNA can have adenine. I put a guanine, that means RNA can have guanine. So we are trying to make it the diagram a better diagram by showing, yes, all of this is possible. But all three uracil also correct. Now you should have an idea. So someone asking me, any living thing got RNA? Every living organism have RNA. No RNA, the living cell cannot survive. Every cell has DNA. Every cell has RNA. Maybe there's, if you take red blood cell for example, red blood cell does not have nucleus. If they don't have nucleus, generally we can say they don't have DNA, but they will have RNA. Why? We haven't gone to the function. Once I explain the function, you'll have an idea why they have RNA. So we'll come to that later. Thanks for asking. Every living organism, every living cell will have DNA, RNA, generally will have. Any question in here? For the moment, the takeaway from this diagram is RNA is single strand, satu rantai. We use the word strand, we don't use the word chain. Single strand is a better answer. So it's a single strand. It has a ribonucleotide. It has adenine, guanine, uracil, cytosine. How do I show this is adenine? Pointed. Rounded is guanine. Uracil is pointed inwards. All of that, you're okay. So now RNA finished. Let's go to DNA. So this is a polynucleotide. Many nucleotides linked to one another, already understand. Okay, now this is RNA. RNA is single strand. This is DNA. DNA is double strand. If I draw, this is one strand, satu rantai. That's RNA. And actually, if I focus inside, if I zoom in here, these lines that you see, the yellow color, this is yellow, orange color, yellow color, green color, all of these represents nitrogenous bases. So this is nitrogenous bases, and you can see that they don't have a pair. This side, DNA is double strand. What we mean double strand is, this is one strand. If I follow, the red one is one strand, and this green one is another strand. The two strands are coiled into one another. When they coil into one another, they form what we call as double helix. You want a better double helix, you can see it in the next page. Yeah. This is double helix. This is what we call as double helix. Clearly you can see here. So this is DNA, this is double strand. Where is the double strand? You can see one strand here, and there's another strand here. So this is double helix. Okay, no problem, Sneha. No problem. Okay, so this is what we mean by double strand. So I go back to my earlier diagram. I'll finish this off quickly. So RNA is single strand, DNA is double strand. And when you have double strand, kalau ada dua rantai, you need something to hold them together. So when there is two chains, you need to make sure they are holding one another so that they don't separate. So that is where our nitrogenous bases comes in. This one doesn't form double helix. This one doesn't form. If it forms also, it is single helix. This one forms what we call as double helix. The shape, the coil shape is helix, and there is two chain, meaning it makes double helix. Difference between DNA, RNA, the differences you RNA ribonucleotide monomer, DNA deoxyribonucleotide monomer, 1 mark. RNA has adenine, guanine, cytosine, uracil. DNA has adenine, guanine, cytosine, thymine. Second difference. Third difference, RNA single strand doesn't form double helix. DNA forms double helix. Why they can form double helix? Because they have two strands. So they are coiled to form double helix. How do you draw DNA? This is one nucleotide. If I want to draw a DNA, what is the specific name of this nucleotide? Can you write in the chat group? I'm going to draw a DNA. So this is a nucleotide, yes. I'm asking this whole thing, this whole thing. This is a nucleotide, general name. Most of you have the answer good. One or two students giving me a different answer. This is a nucleotide. Like my earlier picture, I wanted to make RNA. To make RNA, I needed to use ribonucleotide. Now I'm making DNA. So if I'm making DNA, what I need deoxyribo. So this is deoxyribonucleotide. Why? Because I want to make DNA. Same like what we did earlier. We have many one, third one, and they all form a link. This is one chain, one strand. And based on the pentose sugar, you can see like the pentose sugar is pointing upwards. This one is pointing upwards. We say is in this direction, is pointing upwards. The other side, we need to draw another. The one you draw on the other side must be in the opposite direction. Meaning, if this is pointing upwards, this side downwards. Yes, the name, some of you are giving me the name. I'll come to that. So, what do you draw on this side? You draw them like this. So, there are two polynucleotide strand, but the second strand is in the opposite direction. So this is how you draw the other side, the right side. Pointing up, this is pointing down. Now you can automatically see the importance of complementary pairing here. Adenine must pair with thymine. Guanine must pair with cytosine. Thymine must pair with adenine. This chain, the blue color is facing down. I'm just using red and blue to show you is two different strands. They are just showing is two different strands so that you can clearly see one is pointing up, one is pointing down. Both of them are parallel, serrally, parallel but opposite direction. So many of you already giving me the name. The two polynucleotide strands are arranged anti-parallel. This is what we mean by anti-parallel. So these two strands are held, dipegang bersama. What is holding them together? What will make sure these two does not separate? Between adenine, thymine, between guanine, cytosine, between, we call this nitrogenous base pair. Between nitrogenous base pair, they form hydrogen bond. We call this complementary nitrogenous base pair. You already know adenine must pair with thymine. When adenine pairs with thymine, they form two hydrogen bond. We show hydrogen bond by using broken line. I already explained in water explanation. Hydrogen bond, you don't use straight line, you use broken line. A and T forms two hydrogen bond. G and C form three hydrogen bond. These bonds, it what holds the DNA strands together. So sini ada dua hydrogen bond, sini tiga hydrogen bond. All of these hydrogen bonds holds the two DNA together. So my summary now is, I'm going to use the picture that I see here to make the full definition of DNA. DNA consists of two polynucleotide strands. DNA consists of two polynucleotide strands arranged anti-parallelly in double helix shape. DNA consists of two polynucleotide strands arranged anti-parallelly to form double helix. I think you can write that in your notes. Write this, what I'm writing now here. DNA consists of two polynucleotide strands arranged anti-parallelly in double helix formation. Okay, I need you. So we are almost at the ending of the class. DNA consists of two polynucleotide strands arranged anti-parallel, parallelly. Okay, anti-parallelly in double helix formation. That is supposed to be the last explanation for my DNA. They form double helix, but inside the double helix there is double strand, but they are anti-parallel. Holding them together is the hydrogen bond. Okay, one of the students, I get this question often. One of the students asked me, so why 2 and why 3? There is reason why 2 and why 3. There is a reason, but it is not in your syllabus. Okay, someone asked me earlier why 2, why 3. The answer is here. You can see thymine, adenine, there is 2. Cytosine, guanine, there is 3. I repeat, this is too complex for your level. Take it as 2, take it as 3. Kenapa 2, kenapa 3? It is something to do with the functional groups. If I start to go into that, that's going to take another 20-30 minutes. End of the day, you don't even need it for your exam. So for the moment, accept it as 2 and 3. Yes, there is a reason why 2 and 3, but learn that in Form 6 or A level. When you go to that level, we'll give you the reasons. One of the students that asked me, Mian, I think Mian Hasib. Another student's question is, 1 minute, I change back the slide. Go back here. So another student's question is, can it break? What do you mean, can it break? The it refers to what? I assume you're asking about hydrogen bond. Yes, the hydrogen bonds can break. Is that the question, Surya Kumar? When you use the word it, is it a hydrogen bond? The student asked me, can it break? DNA break. Yes, DNA can break. You increase the temperature, it can break. There are some enzymes that can digest the DNA also. If it breaks, the cell nucleus is the control center of the cell. Inside the nucleus, we have chromosome. Inside the chromosome, we have DNA. So DNA is the one that controls the whole cell. If the DNA already destroyed, the whole cell cannot function. Something like is the brain of the, we have brain. If the brain is not functioning, the whole human cannot function. The DNA is like the brain. We always say nucleus is the brain. Now we are making it more specific. DNA is the brain of the cell. If the DNA is destroyed, the whole cell cannot function. Another student asked me a question. When the DNA hydrogen bond breaks, will the DNA become a RNA? I repeat the question. If the hydrogen bond in here, all of this hydrogen bond breaks, this and this is going to become single strand, correct? Because holding them together is this hydrogen bond between the base pair. If the hydrogen bonds break, that means the double strand is going to separate to become single strand. So the student asks, if the hydrogen bond break, they become single strand, do they become RNA? I'm asking the question, I'm forwarding the question to the rest of the students. Will it become RNA? You tell me whether will it become yes or no. If you say yes, give me, if possible, give me no, also give me the reason. See, there are some students very smart. They don't say yes, they don't say no, it depends. So that means whatever I answer is right. They are on the safe side. Or the students that don't give me the answer, and another student here, maybe question mark. See, she's not right answering the question and she's not denying the question. So right in the middle, play safe. Yes, many of you are giving me the answer that I need. Okay, some said I don't know. Okay, the answer is no. When this, yes, most of you are giving me the answer that I need. Thank you. I'm very happy that you all understand, because this is a very complex explanation, and good that many of you understand and can give me the answer, which is these double strand, when they separate, hydrogen bond, yes, they separate. But what you have inside is thymine. You see a thymine here, you see a thymine here. When they have thymine, it can't be a RNA. It's a DNA. It remains as a DNA, but it becomes a single strand DNA. That's all. But it will remain DNA because it has thymine. This is the answer that someone asked me earlier. Can the DNA become single strand, double strand? Someone asked me earlier. There was a similar question. Yes, DNA can be single strand. Before you can form a double strand, it needs to form a single strand first. DNA can be a single strand. When they bind two, then they become double strand. But they exist in the cell in the form of double strand. They don't exist in single strand. But before I can make double strand, I need to first make single strand. RNA doesn't have thymine. RNA has uracil. Correct, Abhirami? And many of you are giving me the answer. Any other question about this diagram? Just 1 minute. I'm saying 1 minute, 1 minute. I'm taking a lot of your time. Very sorry. There is a partial question. Okay, another good question from one of the students. Why can't the RNA make double strand? The double strand DNA function, which we are going to explain in the next class, it's related to the function which is the DNA stores genetic information. DNA stores genetic. I repeat, one of the students' question, Kalpana, she asked a very important question, which is, why can't the RNA become double strand? Why not? They don't become double strand mainly because it's related to the function of the RNA and the function of DNA. DNA always remains inside nucleus. They store genetic information. I'll give that explanation next week. DNA remains in nucleus. They don't go out of nucleus. They store genetic information. RNA, there is 3 different types. DNA only got 1 type. RNA got 3 different types. And each one of them have different function. To do the function efficiently, they need to remain as a single strand. Kalpana, your question, I need to explain more. We are limited time. You'll understand the answer for the question better next week when I start to explain the function of RNA and DNA. For the moment, they are not, because it is not going to help them in their function. Another student asked me a question. Can the uracil form a bond with adenine? I ask you again. Is the shape complementary? Uracil is pointed inward. Thymine is pointed outward. Yes. Uracil can bind with adenine. Can. But the problem here is RNA is single strand. To form a bond, you need double strand. That will also answer the question that Kalpana asked earlier. You want to join complementary, you need 2 strands. RNA has single strand, so they can't form the complementary pair. To form complementary pairing, you need 2 strands. RNA has only 1 strand, so you don't have the other one to form a complementary pairing. But if is present, adenine can pair with uracil. Can? They are complementary. Another very good question. I'm very happy that all of you are asking questions, and sorry for some of you that I'm dragging the class. If you have something to do, go ahead. You can log out. If, I'm just continuing a bit more since there's questions are coming in. Is it only humans have the 4 type of nitrogenous base? Actually, there's 5 types. There's 5 types of nitrogenous base: adenine, guanine, cytosine, uracil. There is 5 types. Is it only human have? Every single living organism in this world have the same 5 types. Every single one of them. From the largest to the smallest. Virus have them, bacteria have them, fungus have them, amoeba, paramecium have them, plants have them, even plants. And we animals have them. It's universal. When I ask you, you and bacteria, are you all same? Or you and virus, are you all same? Yes, we are different. I'm different. I'm larger size. Virus is smaller size. But I repeat, if you look at a virus, you can't even look at a virus. Let's say I talk a plant. You look at the plant and you look at yourself. You are an animal, a plant is a different organism, but the plant is made of cells. We are made of cells. Inside their cell, inside our cell, we have nucleus. Inside their nucleus, inside our nucleus, we have DNA. Inside our DNA, inside their DNA, we have the exact same thing. Adenine, guanine, cytosine, uracil, thymine. We may look different, but the language of the cell is same. You take your DNA, you can put inside a plant cell. The plant cell can understand the DNA because the code is same. I think I'm getting too excited. Sorry. Any other question? Any other question? So yes, every living organism same. Any other question? Okay, I just finish off with one last one. There's one past year question. They ask you to draw DNA. They gave you this red one. They didn't give you the blue one. They ask you to complete the diagram. Same structure question. You can complete the diagram. So ini dia tak bagi, dia bagi 3 markah. So how do you get the 3 marks? You draw this pointing downwards, 1 mark. You can show me the complementary base pair, A dengan T, G dengan C. That's 1 mark. Pointing down is 1 mark. The direction is 1 mark. Pairing is 1 mark. You can show me here got 2 hydrogen bond, here got 3 hydrogen bond. That's another 1 mark. The direction, the pairing, and the number of hydrogen bond is where you get your 3 marks. That's DNA. So last thing to do, quickly go to your notes, and by now, everything here you should be able to understand. Maybe I can highlight that next week. But everything about DNA you already know except for this last one. This is the only one I haven't explained. Everything here already explained. Just have a quick look at this. This is page 6. Everything about DNA except for the last one already explained. RNA, I have only explained the first sentence. The rest I haven't explained. Only the first sentence has been explained. The rest haven't. DNA, everything explained except for the last sentence. If you have any question, you can ask. If you don't have a question, I'll see you again in your normal classes, the respective classes. If you have question, ask. If not, we are done with the class. We'll continue the rest in the next class.